Yeah, my name's Keaton Tremble. I'm in the Dentiger lab, which is the mycology lab at the Natural History Museum of Utah. We're also the genetics lab for the museum. And I wanted to kind of give a brief intro to a cool project that's ongoing right now that's utilizing help from citizen scientists all around the world. But I also wanted to give kind of an intro into what are questions and research questions that a modern mycology lab asks, so that you guys have, maybe a better idea of what we do up there hidden away in the dark recesses of the museum. So, the title of my talk is How We're Using Porcini, Boletus edulis, to Understand Why There Are So Many Species of Fungi. So, as you guys are entirely aware, the diversity of the fungal world is profound. Current estimates are that there's somewhere between 2.2 to 3.8 million species of fungi. And I mean, that estimate changes, wildly. I've seen estimates up to 5.5 million. Along with being really diverse, we see a ton of different structures and things that look very different that are really closely related. So, I mean, just this screen alone is the diversity of things that are found within a single family, the Boletaceae. You have things that have really loose pores and almost truffle-like organisms. And, this kind of this exemplifies the problem that we have in the mycological world. There's so much diversity and everything looks really different. But we have very little understanding of what's driving this. Why are there so many different species of fungi? So this whole project is to build a system to kind of answer this question of why do we see so many species? So quick overview, I'm gonna give you a quick introduction into ectomycorrhizal fungi. I'm sure you guys know all about it. But then I'm going to hone in on our specific system, Porcini, the king bolete. And then I'm going to give you an example of some preliminary data on some stuff that we've collected in the Uintas with help from citizen scientists and from around the world. So nothing to me exemplifies, this question of why are there so many things out there than ectomycorrhizal fungi. So there's approximately 20,000 species. That's on the very low end of estimates, but it's a really diverse group across, the mushroom-forming fungi. And it is a dominant mutualism. So these are things that associate with trees, and they, help trees. And I'll go into a little bit more of what's going on there. But it's a dominant relationship that is, present across the entire northern hemisphere. But we see lots of hotspots of diversity. So, for example, in, the Western Congo, we see massive amounts of ectomycorrhizal diversity, as well as in Papua New Guinea and other places such like that. So ectomycorrhizae are fungal taxa that live in the soil. They form big nets of mycelium, the body of the organism itself, and they colonize root tips of trees. And that's what this actually looks like below ground. So these two pictures below here are what an uncolonized root tip. So if you went and dug up some pine tips or something from aspen, it'll look something like that. But these mutualistic fungi will colonize and they'll form these structures around the root tips itself. And this structure itself is like really the basis of this relationship. So what's actually going on is that the ectomycorrhizae are really good at uptaking nutrients from the soil. So primarily inorganic and organic nitrogen and phosphorus. But there's many other things that we really are just now beginning to understand. So the mushrooms and the mycelium uptake these nutrients from the soil, and through these little structures here, they provide it to the trees. And in exchange, the tree gives them energy. So trees, photosynthesize, they make a ton of energy. And this energy is, it's a lot easier to acquire for these mycorrhizal species than if they're going to degrade wood and stuff. So we have this relationship. So the mycorrhizae uptake nutrients, they give it to the tree, and in exchange, the tree gives them energy. And it's a really important relationship for the tree. So for example, it's like 15 to 28% of all the energy made by a tree is provided to the mycorrhizae. I mean, imagine if 30% of all the energy you consume was used for little helpers, little, mutualists. Like, this is a really important relationship for both the tree and the mycorrhizae. But what is interesting about this is, even though there's 20,000 species, for the most part, we think everything is kind of doing the same thing. And that makes no sense. How do you have so much diversity if everything is basically doing the same thing? There's some nuances. So, the tree can actually be choosy of where it gives its energy. And something called a market resource economy occurs. So where, the amount of energy provided to the fungi is directly, approximately proportional to how much nutrients it gives. So things that are really good at picking up nutrients and are really good mutualists give a ton of nutrients to the tree, and in exchange get more carbon. But this is basically the extent of what we understand, about these systems. So there's 20,000 different species of things kind of all doing this same thing. It makes no sense. And so this is a really simplified system we see here. We see two different taxa. It can make sense where one provides a little nutrients, one provides a lot. But this is not what mycorrhizal communities look like. So I won't really go into this paper, but this is just such a cool picture. So this little plant here, Bistorta vivipara, is a small alpine forb. It's like 10 centimeters tall. It's tiny. But some hapless grad student in Norway had the task of going in and picking every single root tip from this entire plant and sequencing and trying to identify which fungi are colonized on it. And what they found is, on this tiny little plant, there was over 30 different species of fungi associating with this one plant. And not only that, it's a really complex community. There's spatial structure. Things like other things. Things don't like other things. And this kind of highlights how little we know about mycorrhizal communities. You might see something like this if you went and dug up a Doug fir up in the Uintas. Many, many things are associating with it, but we don't know what they do differently or why there's so many things there. So, to summarize, for ectomycorrhizal fungi, there's a vast global diversity. They form really complex communities in these small spaces. And for the most part, we have only a basic understanding of why they're all there. We really don't understand. So how do we get around this? How do we have any idea, and how do we figure out how and why there's so many taxa out there? So this is where Porcini comes in. So by Porcini, I mean, the king of the clade, Boletus edulis. This is one of my favorite pictures of my lab mates, Alex, up in the Uinta. Just a beautiful, beautiful Porcini. So as you guys know, there's many, many things that are called Porcini or similar to Boletus edulis. And this is a map of what everything from all collections and observations across the world. This is where people are finding things called Boletus edulis. So it's distributed basically everywhere in the northern hemisphere. And just because there's not a dot doesn't mean it's not there. It just means, there's not many people reporting data in Siberia. But yeah, so across the globe, people are finding something called Boletus edulis and reporting it. And, is this one species? Is it not? It's really hard to say. And as you guys, I'm sure, are aware of, there's many different varieties of quote unquote Boletus edulis. We have something called rubriceps, which has a really distinctive red pileus, a cap that is found in the Rocky Mountains for the most part in Colorado. We have a classic Boletus edulis from Utah with a really distended stipe. And then we have Boletus edulis clavipes that is found in the Midwest and out east. And then the last variety, big variety that's found in the US is Grand Edulis, which is primarily found in California, associated with Bishop pine. And this is just the varieties that are found in the US. And, you know, this kind of highlights how much diversity is within this one group. You know, people call Edulis different things everywhere. And this doesn't even get into the varieties that are found in Europe. The Russians have supposedly 30 different species of Boletus edulis, you know. And this doesn't even include everything that's called Porcini. So Boletus edulis is up here, but there's many different things that are, quote, in the Porcini group. It's really complex, and, you know, there's just a lot of diversity out there. So I think what really emphasizes this point is this quote from Alex Smith and Harry Tier. Oh, my bad, sorry. Harry Tier is like two of the most prominent mycologists from, you know, this last century, and they published what is probably still considered the book on boletes. And they said, no matter what species concept one subscribes to, Boletus edulis is a collective species. You know, Singer recognizes as subspecies, many of those as we regard as species. And this is the complexity of Porcini, of edulis, you know, depending who you talk to. Rubriceps might be another species. Clavipes might be its own species. And, you know, I'm going to get to a point at some point, I promise, but this is kind of, you know, identifying that within this group, there's tons of diversity, tons of genetic variation, and nobody's quite sure if it's all one species or not. So, you know, in the modern era, we can sequence. So we can take DNA out of genes and find out differences between that gene and two different samples and see if they're related or not. So we've done this in Edulis, and we think, oh, this is going to solve the problem. We're going to figure out, you know, whether or not it's all one species or yada yada. And it turns out it's really hard to understand. So these are all sequences from stuff we found in Utah. So across this family tree, they're found in different spots. So we've got some up here, some in the middle, some down here. And that's kind of the same thing when we look at East Coast samples or Europe. And what this tells us is that there's no population structure, at least when we sequence this one gene in Edulis. So if all of these varieties are in fact a good species and entirely different, we'll see that stuff from California is a different group, stuff from Utah is a different group, but that's not what we're seeing at all. And when we sequence another gene just to confirm and see if they're different or, you know, telling us the same story, it's complete junk, you know? Nothing agrees, nothing corresponds here. And so this is kind of telling us that, you know, everywhere people are calling Porcini and Edulis different things, but it might be, you know, the same species, it might not be. It's really complex. It's, you know, it's really hard to understand, which makes it a really, really cool system for us. You know, it's a beautiful, perfect project in the making. So the big question really is, you know, is Edulis one species or is it two? You know, is it up here on the family tree or has it, boom, diverged into two or three different species? So this is the classical sense of how species form. You know, you're one thing one day and then boom, you're two things or three things the next day. In reality, speciation is fuzzy. It's this big kind of gray area. And from all of this data and all of this confusion that, you know, we've been reporting on for the past 50 years, we think Porcini is, boom, smack in the middle. It's not one species, it's not two species, it's somewhere in between. And this is exactly why Porcini is a perfect, you know, organism to understand how species form and why there's so many species, because it's in the process. It's in the process of speciating. So this kind of sets up the groundwork for the study. We're going to take mushroom specimens from all over the world and try and figure out, how do species form? How do populations form? What is driving, you know, the massive amounts of diversity we see all over the world? So we're finding eduous specimens in museums from across the world, and we're taking DNA from these specimens, and we're not just sequencing 1 or 2 genes, we're going to sequence the entire genome of all of these organisms. And hopefully by the end of this, we're going to sequence over 600 genomes from across the globe. And I want to take a moment to emphasize, the importance of fungaria and collectors across the world and natural history museums. Because without this resource, without all of my partners across the world, I've got a museum in Siberia that has sent me stuff, and they get their stuff from amateur collectors who bring stuff to their natural history museum. I've got stuff from Iceland, New York, Denmark. Without this really complex system of collaborators, of amateur collectors and citizen scientists finding stuff, reporting it, and storing it in natural history museums, I wouldn't be able to collect Porcini from across the globe. So we're gonna take all of these specimens from across the world, we're gonna take their DNA, and we're gonna sequence their genome. And what does that actually look like? What does that even look like? So I'm gonna skip that. So what can we do with all this DNA? So across the genome, we can look for mutations. Oh, that's actually a good point. Little bits in your genome that are different than anything else. And we can quantify that and use that to, put things in populations and see how different groups of Porcini and Eduus, are changing across the entire globe. So this is preliminary data from what we have right now. And we see that even though there's tons of diversity in the West Coast, everything is kind of one population. So Grand Eduus is probably just Eduus. And, the East Coast is its own population as well as Europe. So whether or not these are 1 species or 2 species, it's kind of hard to tell at the moment. We'll figure that out. But, from simple data like this, just putting things in a population, we can figure out really cool, make really cool hypotheses. So for example, based on this data, we might be able to say something like this, that Porcini arose in Siberia. And what we see is that a population has moved into Europe and a population has moved into the West Coast. And from that population, something has moved to the East Coast. And we can track and measure how these species, move across the globe from this genetic data. And this will really give us an understanding of how, species form, how fast they form, when do they form. And we can take this one step further with this data that we have. And this is all data collected from museum specimens that have been stored in natural history museums, for decades potentially, collected by someone 50, 60 years ago that we've requested and has been sent to us. So we can go a step beyond this. So we can say, hey, these populations are diverging. What we think is Grand Eduus is actually, probably Eduus. But we can try and figure out what environmental factors are driving, divergence. What environmental factors are making that its own population or this its own population. And we can find, genes under selection. So we can, across the entire genome from these mushrooms, pick out little mutations and see what environmental factors are driving those mutations. We can ask, hey, is this mutation associated with elevation, precipitation, temperature? So from this one species, we can get an incredible amount of data, and we can form a lot of hypotheses about why are there 20,000 different species of mycorrhizae? Well, some may be adapted to temperature. We actually don't know if mushrooms adapt to temperature or precipitation. It seems obvious, but it's really actually hard to find out if precipitation makes new mushroom species or not. So at the end of this, we're going to collect Porcini from across the world, and we'll be able to figure out how do species form, how fast do they form, how fast do population forms, and at what scale are these populations? Is something that we find on the West Coast its own population, or is the entire continent of North America These are all questions that we have no answer to so far in mycology. And then we're going to go a step beyond that and see, hey, for these Tylopolis species, maybe they're adapted to elevation or nutrient content or something like that. So, the humble porcini that you're finding in your grocery store is really going to be the resource. And it's going to really help the entire field of mycology understand why we have 2 to 3 million species. Because right now, it's kind of up in the air. So I wanted to keep it short and just give a quick overview of what we do. So I want to thank my advisor, Bryn, my lab mates, Alex, Bryce, Dally, Abby, Jacob. And I can't, thank the Natural History Museum enough. Like, working in a Natural History Museum is a very special thing. And being able to access these collections that have been collected over 100 years ago, maybe, from around the world is something really special. And those organizations don't work without the help of citizen scientists, finding specimens and stuff like that. So that's all I got. If you got questions, I can answer them, or else we can move on to identifying what we found outside.
Thanks, Keaton. That's fascinating. I know I have questions. I don't know if everybody else does. I was wondering, you mentioned that ectomycorrhizal associations, you kind of had the distribution plot, and you said it was all over the northern hemisphere, and you had very few of those in the southern hemisphere, as well as the Boletus sightings or observations. Yeah, very, very little, a little bit down in southern Africa. Mm-hmm. Is that because of a lack of data, or is it that we think that they really, those species don't exist, like Boletus maybe doesn't exist in almost all of South America, and maybe ectomycorrhizal associations in general are less common?
So ectomycorrhizal fungi primarily associate with, it depends on the group, but for the most part, they are not a southern hemisphere group because many of the things that they associate with aren't distributed through most of the area. But Chile does have a really high diversity, as do, it's mostly temperate forests that they're following. And with almost all southern hemisphere stuff, we just really don't know what's down there. So the edulous dots you were seeing on that map are actually probably introductions. So as mostly European settlers, we think, move around the world, they've carried porcini with them. So, and we know they're introductions because they only associate with introduced trees. They're not really associating with native trees. Like the biggest example is New Zealand. So New Zealand has no native conifers. So all of the edulous we found out there is associated with Doug fir people brought over for timber. But it looks like wherever European settlers went, they took porcini for the most part. But yeah, so the reason we don't know much about ectomycorrhizae in the southern hemisphere is nobody collects down there. It's really hard to get to, and it's a shame because that's probably where a lot of the diversity is.
Thank you.
I got a question, if I can ask. Does it hold for mushrooms as it does for other plants and animal species, that if you got 2 Boletus and you suspect they're the same species, but you crossbreed them, do they come up with hybrids that are sterile, or are those hybrids, prolific?
So that's a really good question. So if they're the same species and they're compatible, so mushrooms don't mate in the same way that plants or animals do. They have little spots in their genome that say, yes, I am compatible or I'm not. It's pretty simple. But we really do think that they can hybridize, and evidence is really, really limited. So it's kind of hard to see since it's really hard for us to tell if something is one species or not. But we think this is pretty common. And we see, at least in porcini, we see examples of things from Europe hybridizing with stuff we find out here. Whether or not that's someone carrying a spore or something like that, or it's just really long distance dispersal. We know that things can be separated for quite a while and still reproduce and make, viable, I mean, again, whether or not they're viable is a really hard thing to tell. They might make spores, but are those spores crappy? We don't know. But we think it's pretty common. We think they can hybridize between, sister taxa and stuff like that.
Good answer.
Awesome. Well, thank you so much for your time.
Allison's raised her hand, I think. Allison?
Oh,
Hey, Keaton. So I was just wondering, why Porcini specifically? Is it its distribution, its identifiability? Like, why Porcini rather than like a Morel or something else?
so a couple things. One, it's an ectomycorrhizal taxa, so it's something that I'm really interested in. But everybody loves Porcini. Everybody loves Porcini, no matter where you go. The Russians really love, things called Boletus edulis. And because everybody loves them, everyone collects them, and it has a huge distribution in natural history museums because everyone collects it for the most part. And it's really easy to find people to collect. So in areas where I know there's edulis, but I haven't found something in a museum, I can go on Mycoportal and try and contact someone who's posting pictures of Porcini. Yeah, because everyone collects it, it's really easy to find specimens from everywhere. Why specifically Porcini? I think it's the whole species complex, that I talked about earlier, because it's really hard to tell if it's one species or not. It's in the progress of becoming two or three species. So we're looking at it in a really good snapshot in time to see, the genomics behind the process of speciation and try and identify how this process actually happens. And I love eating it, so, it's really easy to get me motivated to go collect edulis wherever I travel.
So Stephanie here had a question about if those you talked about the distribution starting in maybe Siberia and moving out to the other places.
Mm-hmm.
Do you think that's following human population movement, or is it maybe separate or just natural dispersal?
It's definitely natural dispersal.
And she's saying also, do you
have a time frame for the distribution?
Time frame for the distribution? Are you able to figure out when these distributions happened?
So I don't have a time frame yet. We need a lot more data since we only, have a few specimens from all over the world. It's really hard to say with any certainty, like this population has been diverged for, 1 million years or a couple hundred years. But I think the distribution was probably here before humans. It's what it seems like based on, how diverged these things are. A lot can happen in 10,000 years, but these things are, relatively different. So I think it was here before humans. And we know that, it's rare, but studies have shown that spores can disperse between continents. Whether or not that, is a big part of actual gene flow, but we know it can happen. So we know that these rare colonization events may occur. So what actually kind of looks like is happening now is something called like a post-glacial migration. So as the glaciers and the big ice caps came down, 12,000 years ago, it really cut off these populations because there's no dug fir or hosts or trees between these big populations. So it kind of looks like as the ice has receded that these populations are expanding again. And, I have a couple specimens that appear to be hybrids or maybe a new population that are centered in Alaska and are coming down British Columbia. So that might be Siberian edulis moving over again, or it might be Alaskan edulis that's its own population and is slowly extending out again. But yeah, sorry, a roundabout way to say I think it was here long before humans, but we don't have an actual date yet.
Great, thank you. Other questions? Nothing? Well, Keaton, that was fascinating. Thank you so much for sharing your expertise and your current projects. So, how long have you been working on this, and how long before you feel like you'll be done with answering some of these questions anyway, or this project?
Yeah, so we've been working. So, I mean, Bryn is the Porcini guy in the world. He is known as the edgeless guy. So this has been his baby, for a decade. But taking it to this next level of doing whole genome sequencing, that's where that was my drive and what I wanted to get out of, my PhD. So I've requested over 2,000 specimens from around the world from all of these museums or people I find on MycoPortal or, people I find on Mushroom Observer. So really it's however fast those come in and I can extract DNA. So I think we'll have the world's largest genomic data set of a single species within a year, and we'll be able to start answering some of these questions. But the cool thing about this data set is you can ask anything. Once you have this data, there's so many different things you can ask, and it'll be, if I wanted to, it could be a never-ending lifelong project. I will be done with it long before then. But I hope to answer some of these basic questions within probably the next year.
That's cool. Well, thank you, Keaton, and hopefully all of us can spare a Boletus here and there when we're getting stuff.
Oh yeah, especially if you're in the Tushars or the La Salles or any of the mountains down south. We know they're there, but we just don't have samples of them. We have many, many, many from the Uintas, so I appreciate if people wanted to bring in some Uinta specimens, but I think we're pretty good there.
So everybody got that? If you get Boletus up in the Uintas, just take them home and eat them. They don't.
Yes, you were going to do that anyway, but.
Besides the La Salles, what was the other range you said?
So any of the ranges in southern Utah that have conifers on them, so the Tushars, or even down at Cedar Breaks, I haven't been able to find any edgeless down there. It's definitely down there. I've just only hit it at dry times. So. Yeah, or especially any of the ranges out in Nevada. That would be really, really cool.
That's where we were.
Yeah, we'll see what we can do as a club also to facilitate that and get a few specimens from down there. That'd be. We should be able to arrange that.
I will say, so some of the specimens you guys collected last year's Foray are in this data set. So they have genomic sequences and, they're tagged with the collector's name and they'll be in this data set forever.
Cool.